Welcome everyone to the flagship episode from Code to Cure, Ainnocence webinar series exploring the intersection of AI and biotech. Today's episode is titled AI in Drug Discovery: What's Real, What's Hype, and What's Next? I'm Kevin, and I'll be your host for this session. Today, we're joined by Dr. Pan, the visionary founder and CEO of Ainnocence, a company at the forefront of AI-driven biotech innovation.
Hey, everybody. Very nice to be here. And this is the first episode of our webinar series in the hot topic of AI in life science and pharmaceutical industry. So I will be sharing all I know about AI and its application in this field without any reservation. Thank you for your attention, and I hope you enjoy the talk.
Okay, great. Let's start with a quick overview. For those who might not be familiar, what is Ainnocence and what problems are we trying to solve?
Ainnocence is actually has a metaphor of innocent AI, as we aim to use AI technology in the good cause for a good human society and also for the benefit of human health. And we're building this company in order to leverage the advanced AI technology, including advanced algorithm and also the vertical field in the life science and data science to be able to expedite the drug discovery process and also tackle some of the scientific challenging in drug design and to make some really breakthrough therapeutics for some incurable disease that so far. And there's about 1,500 diseases without treatment currently. So we definitely have a lot of work to do to eventually eradicate those human sufferings.
Yeah, that sounds very interesting. And I heard a lot of very big keywords in the opening statements. And so let's start by cutting through the buzz. AI and drug discovery, it's been a hot topic over the last few years. So let's explain this little by little. What's actually working today, and what are some real and proven use cases out in the market right now?
Yeah, definitely. So this is drug discovery, and it is a very big, long process that involving multiple process that with different disciplines from chemistry, biology, and pathology, and physiology, and also other medical professions, background knowledge in order to push all the necessary research from the beginning to the end. And traditionally, this process normally would take years from the screening of the drug to all the way to a clinical approval. And if we just simplify the entire process that probably involve hundreds of steps, there are three major processes that is impacted by AI, and actually we already see some success stories. The first one is in terms of the research of disease biology and be able to extract information from multi-omic data of the patient and be able to identify therapeutic target that is relevant for the disease. So this field has been having many novel discoveries of new drug target in the past 10 years. And also the data that in this field is also enormously big that covers very large patient sample, and each patient sample has multiple layers of data that contributes to that final conclusion between target and the disease. And so this is one of the major application. Then the second step is once you identify a putative therapeutic target, we will use AI technology as well to do drug design. And drug design in the past was either done very manually or have done using very traditional software for very large scale simulations that consume very large scale of computational resource that's probably equivalent to the cost of wet lab screening. But nowadays, we use AI to be able to leverage the history of the pharmacology research and all the data involved in the past molecules that has been tested, so that we'll be able to build very efficient and accurate AI models to generate novel molecules effectively and be able to validate them rather quickly than the conventional blind screening, which would involve like millions of molecules being actually tested. Now we only need a few or just dozens of them being actually tested in the wet lab. And then the third step will be the clinical trial. So nowadays, AI technology has been heavily involved in patient selection and also clinical trial design and execution, and also clinical data analysis. So make clinical trial really effective. We see many companies that involved in this field, even make clinical trials that in multiple regions completely automated and with intelligence management systems, and also the data extraction and also an analysis system as well. So I think all those like processes has been very quickly adapt AI technology in the past 2 or 3 years. And Ainnocence primarily focused on the drug design process, which is a core process that is the AI technology applies for generates the core IPs of those therapeutics. And we have done over 60 different therapeutic discovery cases successfully with our wet lab partners and our clients, which are pharmaceutical companies that has the drug discovery projects. Yeah, we do see like just the recent probably 2-3 years, this field is having much knowledge-wise really has a lot of breakthrough, and also the validation has been more and more convincing, then that people start to adapt this type of technologies more widely.
I definitely read papers in the past, and over the past few years, there's definitely been quite a few papers that have been validating the use cases for their AI models for some of these use cases. And so some of these real world applications include like molecular generation, structure prediction, and also hit identification. So can you give us a brief introduction about the second part of the drug discovery process and how AI is used for these processes?
Yeah, definitely. So first of all, some of the terms such as heat generation, or we say like the starting point of identify a drug candidate that might be has therapeutic effect. So the hits, which means it has showing biologic efficacy through certain in vitro assay. And the AI is currently be able to first generate very large chemical space up to like billions of molecules in a few hours. And at the same time, the AI models are able to select among all those novel chemical space, those ones that are relevant to the target we're interested, and also relevant to the pharmacological profile that the disease that we're interested. And normally this generation process also combined with a multi-parameter optimization process to make sure those molecules are having multiple characteristics that satisfied as a drug candidates. And normally the process goes with screening billions of possibilities and only test a few in the wet lab. And conventionally, let's see, a blind screening for any random chemical space is about 1 out of 10,000 molecule. And then the first generation of computer, a drug design is about 1 out of 100. And then the current AI system could achieve as much as like 3 out of 10. So it's a really improve the hit rate so that we can do much less like wet lab experiment. So this is the hit generation process and how it goes. And then for the other, the next we will normally do like lead optimization. And also structure prediction is somewhat important to understand the therapeutic target's function and its drug binding like regions, and how and rationalize how we should design a molecule to interfere its function. And there's already been a lot of achievement in this field. Even the last year, 2024's Nobel Prize was heavily awarded this area that multiple scientists that has been contributing in the field that in the past that a structure of a target takes years in the wet lab using crystallography or crowd EM. Right now, you only need that to the AI model is able to predict it very quickly with the data has been learned. Yeah, so those are also important for us as the beginning point of the drug design. And also the drug optimization process is once we're closer to the candidate, we're also using AI technology to expand the chemical space somehow that is more focused and relevant to that particular drug target, and also that disease that, for example, if we're optimizing a drug that targets a brain tumor, so that we have to build a blood-brain barrier model to make sure all those design molecules can pass the blood-brain barrier. But if we're designing a drug that is targeting, let's say, a kidney disease, we actually do not want it to cause side effect that pass the blood-brain barrier, so that we have to counter-screen the blood-brain barrier. So those are all pharmacological features that we need to consider to further optimize the potential side effect and also efficacy of the drug.
Okay, so yeah, that sounds very interesting. And let's go back to the first part. So you said right now, structure prediction is they've won Nobel prizes. So that was in 2024 for AlphaFold model. And so I want to circle back into this AlphaFold model. So how is AlphaFold used currently in the AI drug discovery space for the real proven use cases?
Yeah, the AlphaFold model, like I said earlier, it is very useful to output this 3D structure of therapeutic target and all the complex of the binded mode between the therapeutic targets and the other molecules, such as the antibody or a small molecule or a natural substrate of that target. And those are very insightful for us to study its structure function relations. And once we have that in hand, we'll be more focused on exactly how the drug is binds to the molecule, and then we'll be able to narrow down our screening or computation focus on that a very particular group of amino acid that contributes to the therapeutic effect. So I think all that is a very typical process and use case that how people use AlphaFold to initiate their drug screening process.
Yeah, yeah, AlphaFold is very useful for our use cases. We can get a public domain protein structure or protein sequence, and we can turn it into a protein structure for use in our AI modeling process. And overall, the results are very accurate. And so when we have compounds to screen, this is a lot of hit identification work. And as you said before, with a random screening, it takes probably 1 in 10,000. And then with traditional CADD, it takes 1 in 100. And now with AI, it's becoming more and more accurate. So based on the accuracy, it's not like in other domains where accuracy is of 90% is pursued. For the wet lab, accuracy of like 10%, 20% is pursued. So what are some of the other common misconceptions you see in this space?
Yeah, definitely. So it's like yeah, in terms of how we measure the success matrix in the drug discovery process, there and also how the model is being performed, good or bad, there's definitely some misconception. For example, in the eyes of a AI engineer, let's see, a model has to be perfect as like maybe 95 or 99%
Yeah.
accurate. That considered to be accurate. Even we can use, different type of matrices that like a Spearman correlation or PM like, and also like some F1 scores or recall scores. So we have multiple matrices that measure how a model is performed, that we know how well it might predict in the real case. But in the drug discovery, interestingly, it's like even if your model is like scored like like the validation testing set is scored as much as 95-99%, it doesn't mean all you predicted in the wet lab can be has the same actual perspective accuracy. And the different measurement is actually we use a hit rate to actually measure how effective this model is. It does not have to be 100% or as equally like 95% accurate. We just need to be better than the traditional methods that's already serve the purpose. And also because biology system is very complex and data might be with noises. So it's not always the case that the model training scores and the testing scores are in the virtual space and completely correlated to the actual wet lab space. So, but in the wet lab space, we still need to use a very rigorous way to measure success, which we would have a separate standard. But like hit rate is something very common. Sometimes the model scores, let's say pretty low, like for example, if they have their like R square of 0.6 or 0.7, that people would think that the model might not work. But in the real case, if it's a very relevant domain that the training set and the predictive set is related, that still could be much better than just blind screening.
Yeah, that answer is very interesting, and I definitely agree with the answer because sometimes when we build models, we have a AUC score or a recall score of over 90%, which is very high in terms of in silico modeling, and this is considered like a very acceptable model for us to use. However, in the wet lab, there's multiple different variables, such as maybe the assay has a slight variation from what the data we used to build the original training set was, or because the target might be a little different than what it's actually in our training set. And so I think that's a big expectation. And however, there's also a lot of overhyped expectations, such as, in small molecules, AI will replace chemists and things like that. So maybe can you explain to us how Ainnocence, we also use chemists, but we also use AI at the same time.
Yeah, definitely. So, first of all, I just all of a sudden thought about that the some of the misconception. I think it's very important. I think there's a common misunderstanding that people think that if training data does not cover the information of a target that you wanted to predict around, the model might not be effective. But actually, in our case, we think the mod, if as long as the base data is large enough and also diverse enough, it has the transfer learning power to even predict the drug candidates for completely novel target that it has never been seen. Because there's underlying patterns in among amino acids, even there are different protein family or there are different size of proteins or different regions. But because we have learned if the AI has been saying so many similar pattern in the regions of a drug binding amino acids crowd, I think it will be in our experience actually it still can generate novel molecules against the novel target, kind of thinking out of box. So I think that's something that most people think that AI cannot do, but it actually can do.
Yeah, that's correct. Because when we build our AI models, so let's say there's around 1,000 different targets in our training set. So out of those 1,000 targets, they're all proteins and it's all 20 amino acids. So there's definitely only a set number of combinations that each substructure of the amino acid sequence or structure will have. So we definitely can transfer the knowledge that we have in the training set and put that to our testing set and send these results into the wet lab and achieve very good hit rates relative to something like the traditional random screening. And so, yeah, let's talk about how we use AI with chemists. And so I think it's an overhyped expectation that AI will replace chemists, but that's not actually the case that we've experienced in our use cases. So maybe you can explain it to us how we use chemists together with AI.
Yeah, that's a good question. And it's also another common misconception. I think, first of all, when we build our AI system, a group of chemists has contributed greatly, giving feedback and to form a reinforcement learning cycle for us that using their empirical knowledge. And also, we have been, during the case study, we kind of iterate between AI generation and feedback and cherry picking from chemists and to generate better results. Because at the beginning stage of AI, it still needs to learn not just from pure data, also some of the chemical rules that is not actually written in its original algorithm. Actually, we have been incorporate over 1,000 medicinal chemical rules that extract from those chemists' experience and also literature, which greatly improved our system. I think instead of like saying that AI would replace the medicinal chemist, it's more like first medicinal chemists to build their knowledge into the AI, and also they use the AI to expand their chemical imagination more efficiently to better finish the task. We still need the chemist to guide the AI to do a better job. So I think it's more work as like augmented reality. Yeah.
Yeah, I think there's another keyword that I'm thinking of right now. It's called knowledge distillation. I think in terms of knowledge distillation, we're actually distilling the knowledge of the chemists into our AI models, and we're using what their data as labels into our AI models. So it's a very interesting feedback loop, and it's very successful as well. And based on the data that we have, I also want to talk about data quality. This is often a very overlooked topic, but it's also very important to have high quality data in this space where data is very sparse and data is very scattered and dirty in the public domain.
Yeah, very good question. I think, yeah, Kevin has go through this process that since the beginning of the company, that so definitely that's the key of our engine is to first build from a very clean, relevant, and well documented and well qualified data set. And to begin with, like this field, like in the field of medicinal chemistry and also biochemistry, cell biology, and later stage of human trial experiments. From since the scale up of the system size, the error is propagated from a few percent of the standard deviation to like a few log of error, range, which is really big. So in order to really leverage the usefulness of this data, we have done very rigorous data cleaning process. There's a few techniques. So first of all, we try to identify experiments conditions that align with each other, and also for the same measurement, if we find a contradiction between different research group and experiment, we normally would also either completely delete them, or we were using certain ways to make a judgment of which data is more reliable than the other. And also when we do most of our models are actually classification model, because we find our regression is not in this, if it's not the experiment is not conducted in the same condition, the regression number doesn't make sense. But normally we'll be able to at least tell the positive and negative data, so that we'll be able to relatively more accurately build our classification model than the regression model. And also we kind of itemized each type of assay and then their relevant data to not mix all the data together. So instead of build like a one huge heterogeneous model, we actually build individual, very homogeneous model one at a time. So to optimize one parameter with one model, very, very accurate, and to using them as a combine to do our final prediction and designing. So we do have like data quality process by using different type of training methods, such as we kind of doing multifold cross validation to make sure all the data has been leveraged very evenly without bias or kind of overfit problems. And we also use different type of splitting methods to make sure, such as we split in the chemical space very evenly, and also we split by like time that those experiments has been conducted, so that to make sure those scenarios apply to the future events better. So there's really a lot of details. Yeah, I can just talk this for the whole day, but definitely this is the most important step of
Right.
how we can build a good AI model. Yeah.
Exactly. Yeah. Data quality is very important to us. And without good
Mhm.
quality data, we don't get the models that we're looking for. So
Mhm.
let's make another segue into where Ainnocence fits into the drug discovery space. So you've introduced the company quite a bit. So what makes our platform different compared to some of the other platforms out in the market? And what is our general approach to tackling these AI in drug discovery problems?
Yeah, very good question. So we actually have adapted a very relatively unique approach at the beginning of the company. Instead of predicting 3D structure or using 3D molecular docking for small large molecule interaction prediction, we take a completely different approach that by leveraging the one-dimensional algorithm called the protein language model, that we directly model a therapeutic target in its 1D space, and then along with the therapeutic candidates also in the 1D space and at the maximum 2D space, so that without extensive 3D coordinating computing. So this approach actually have two advantages by design. The first is there is way more data that is available for sequence information, which is just a name of a target, right? And also its experiment result with certain chemicals or antibodies. Those are much more abundant than a 3D kind of interaction data or a bit of mapping data. So those are what we think it has leveraged much bigger data space, and also directly correlates to what we eventually going to measure, which is in bioactivities. Another very obvious advantage of this exposure is the huge saving for computational cost. Without modeling the 3D dimensional space, we'll be able to generate like thousands or even 100,000 molecule with a single GPU or CPU, in a few minutes or hours, instead of if conventional, for example, FEP calculation for a single molecule would take a day. So it's a completely different level of computational cost. It's at the orders of the magnitude of 5-fold. So just 10,000 times saving the computation cost. And I think those two qualities will make this platform more universally applicable to different size of company and saving the cost, and also widely applicable to different modalities from small molecule, large molecule, or even bigger therapeutic modality. So that's why we think even at the time that we were probably penny and nobody believed this gonna work. But after 3-4 years of multiple case valuation, also academia started to adapt this model and this approach, and had some new publication in the last couple of years, which also aligned with our strategy. So we think the future of this technology map will be very, very prosperous.
Right. And now let's go back to the actual topic. So can you walk us through a use case or success story at Ainnocence?
Yeah, definitely. I think one of the very complex case we have done is for a European research institute that they're developing a antibody for multiple COVID strains. I think they give us a very tough case that they wanted us to de novo generate antibody that can target 5 different COVID strain, and with at the very potent like nanomolar scale potency for all of them. And then they actually at the beginning they don't believe AI can do it, but we generate novel antibody using our platform, against this five different strains 3 times. Each time we give them 50 antibody to test. And then the result is actually quite surprisingly good. So we'll be able to identify, I guess, a dozens of them the first round, and the second round about 20, half of them are all hits. In the third round, we'll be able to generate more hits and with pretty strong improvement of binding affinity and other cellular activity based on the wet lab that they carry out in-house. So we have been successfully deliver the project, and the total cost is our computational time is like about 1 or 2 week each round, and then they just did like 50 antibody experiment trials for 3 rounds. And we were foreseeing this type of situation in the future. If there's a pandemic happen with the completely novel strains of pathogens, we'll be able to actually react as quick as this and to be able to generate a potential therapeutic candidate, clinical candidate within just a couple of months maximum. So that's kind of one of the typical case. We only take the sequence of the antigen or the therapeutic target, and then we'll be able to quickly generate antibody and express them and test them in the wet lab.
Yeah, I think we have a similar
Okay.
case that's actually published, and it's for a similar target. And what we did was we had a wild type COVID strain, and then we also mutated the antibody to target Delta strain and then also the Omicron strain. I think we've achieved similar hit rates as the commercial case as well, but this one is completely in-house and was also published.
Mhm. Yeah, if you're
Yeah.
interested, you can check our publication for more detail for this type of computation.
Yeah, and for that one, the timeline was quite long because from wild type to Delta to Omicron, that took about several years. And initially, we only targeted Delta, but Omicron suddenly hit in early 2022, and then we identified an Omicron strain, and then performed the mutations and got quite successful hits from our experiments. And for that overall experiment and study, it took about 6 months to a year. However, in other projects, some of the specific timelines to the candidate delivery, it only takes 2 weeks, as Dr. Pan said. So maybe, Dr. Pan, can you elaborate on this a little bit more?
Yeah, it depends on the case. It's like infectious disease is quite different. Like the ones you talk about, we actually against about 160, 1,600 strains at the same time, actually, and over the spike protein is actually 64. So that was quite a huge computation. But we do the wet lab testing multiple times. Is we wanted to test if the initial design antibody is able to also neutralize the later emerged strain. So actually that antibody was the first round was already successful within like a couple of months, but we tested the effectiveness like over time and see if it's effective. So it's actually we just want to test if that's its mutation resistance in the future strain, and which it is, which is very surprising. So which means it's actually has a elongated protecting period of this type of AI designed antibody that could protect the population for even longer period of mutational time of the passaging evolution. And for most of the cases, it's like just a human target as for example, oncology target that we just having antibody to directly just effect on one like human and non-mutating target, and then it's very straightforward. We designed it for 2 weeks and then experiment validating another 2-3 weeks, and if it's the first round is good, that sometimes happen, but most of the case the second or third round are needed to optimize for the ideal situation, which would take another 1 or 2 months for each round.
Yeah, so I think overall, if we do 3 rounds, it's going to take around 6 months total. And that definitely speeds up the drug discovery time compared to the traditional methods where maybe 3 rounds will take over a year or maybe even 2 years
For three to
for the
six
discovery
years
process.
sometimes. Yeah. Mhm.
Yeah. Yeah. So based on all of our successful use cases, let's be honest, it's not easy, but we've had quite a few successful use cases. But there are still challenges remaining, and what are some of the toughest challenges that we have seen in this field so far?
Yeah, definitely. I think, for example, for small molecule, is more challenging than antibody because there are two bottleneck. One is the synthesis cost and also success rate of being synthesized. Is very, it's very challenging. So even AI be able to design very promising molecule, first it might not be able to synthesize. Even we cover the synthesizable model, but it could be challenging and also very costly to give feedback. So the cost of synthesize a molecule is probably about $1,000 to $10,000, but the cost of antibodies is $200 to $500. So it's just very different. So people are not willing to synthesize more molecules for small molecule. That will also make the total data size data like less, so that the reinforcement learning would not be probably as effective as the large molecule. And another one is sometimes for novel target, there's a lot of uncertainties that we probably don't know fully understand how to interfere its function. And also some of the new proteins that cannot be fully synthesized or expressed to do some very accurate assay test for the drug candidates, so that there's some noises in those assays because they're not fully optimized, so that either give us false positive or false negative in the wet lab. That could be, confusing the AI, not sure which direction to be further optimized. Yeah, so those are some of the challenges that we met, that's some gaps between virtual computation and the actual yeah, the actual wet lab.
Yeah. And so looking forward, where do you see AI having the biggest impact in the next years? And what are you most excited about, Innocence, for the next year?
Yeah, definitely. I believe a lot of AI design molecule will go through clinical trial in the next few years, and people would see a lot of success in this field that AI is actually design better drug than conventional methods, and give the confidence in the field. And that I think the next big thing about Innocence is like probably some of our collaborative project will go on clinical trial, and at the same time we'll be able to innovate novel molecules at a really large scale that we hopefully finish like 100 project in the next year or two, so that we will have a statistically significant success rate for all our project. And also people will think this is industry qualified methodology rather than just a random success like those happened in the past that people don't know what is actually contribute to the success. We wanted that AI take the credit this time if we have a large sample size for success.
Yeah. I think this sample size, even 50 is enough, but obviously the more the better, the more
Yeah,
statistically significant it will be.
yeah.
And so in terms of the overall pharma industry, do you think they're ready to adopt all of our AI tools or all of any AI tools in the market so far?
Yeah, I think they're ready. I think right now most of the AI tools are better and better. I think the entire industry is starting to adapt it in various of stages of discovery and commercialization.
Okay. Yeah. So overall, it's been a very fascinating conversation. But before we close, do you have any final thoughts for our audience?
Yeah, I just think whoever interested in our platform and this type of technology and field, just give us a like or also try to subscribe our series, and we will give more interesting talk and very 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 scientific and technology and innovation in the field that we access to. And yeah, and also give us any feedback you have that to make this the webinar series more interesting and more meaningful in the long run.
Yeah. So thank you, Dr. Pan. Let's be sure to subscribe and stay tuned for the next episode in our series. And also, if you have any questions, please feel free to email us at any time. Our business colleagues will respond to you as soon as possible. Thank you.
Yeah, thank you, Kevin.
All right. Bye-bye.
Bye bye.